All right, as medical students, you spend a ton of time learning about biochemical cascades, right? Hormones binding to receptors, enzymes triggering pathways, the usual suspects. But today, we're going to explore biology through a completely different lens, and that is physical force. Welcome to this explainer on the mechanobiology of skeletal muscle. We're going to decode the complex cellular signaling pathways that translate physical forces, things like stretch, tension, and pressure, into systemic health. By the end of this session, you'll understand exactly how physical loading dictates not just muscle mass, but literally whole body physiology.
Okay, let's dive right into this. Here's our roadmap for today. First, we'll cover the basic principles of mechanotransduction. Then, the two primary models for channel gating. Third, Piezo1's absolutely crucial role in muscle health. Fourth, ultrasound therapeutics. And finally, skeletal muscle as an endocrine organ.
So, Section 1, Principles of Muscle Mechanotransduction. For anyone stepping into the clinic, understanding how cells actually sense mechanical load is foundational to treating musculoskeletal pathologies.
At the very core of this topic is a concept called mechanotransduction. Simply put, this is how cells convert physical forces into intracellular electrochemical signals. Now, skeletal muscle operates around what we like to call the lazy zone. Think of this as a homeostatic sweet spot. If you just maintain your typical everyday activity, your muscles stay right in the zone. They repair and maintain themselves without any net gain or loss of mass. But what happens when a patient falls out of this zone? Think about prolonged bed rest, or say a patient whose limb is immobilized in a cast for weeks. Well, that sudden lack of mechanical loading triggers an incredibly rapid cellular adaptation, leading to severe muscle atrophy. The muscle literally senses the absence of force. And this brilliantly illustrates what's happening at the microscopic level. Look at the sheer diversity of mechanosensitive ion channels embedded right there at the cell environment interface. You've got the piezo family sensing membrane stretch and acoustic force, TRP channels responding to shear stress and heat, and voltage-gated calcium channels acting as these massive signal amplifiers. These proteins are the molecular transducers. They essentially act as your body's physical antenna. Moving right along to Section 2. Channel gating, two primary models. Let's look at the actual biophysics here. How do these physical antennae actually open their pores?
Historically, there's been a bit of a debate, with two competing scientific models to explain this channel gating. On one side, we have the tethered model. This theory proposes that ancillary structures, like the extracellular matrix or internal cytoskeletal actin, act literally like tiny ropes. When the cell stretches, these ropes physically pull the channel open. And for a long time, this was the favored idea, mainly because eukaryotic cells have such complex cytoskeletons. But on the other side is the force from lipid model. This suggests that mechanical force acts directly on the lipid bilayer itself. Changes in membrane tension or curvature drive the channel to open, no tethered ropes needed at all. And today, modern experimental evidence is heavily shifting our understanding toward this lipid-driven mechanism.
Now, what's really interesting about this slide is the force from lipid model on the left. Notice how in its resting closed state, Piezo1's unique propeller-like blades create this localized depression in the cell membrane. We call it a nanodome. When mechanical tension increases and the membrane stretches, this nanodome flattens out. That flattening exerts a direct lever-like force on the channel blades, pulling the central pore wide open. The lipid bilayer itself is the primary transducer of the force. And the proof for this is just fascinating. We can actually trace this force from lipid mechanism directly back to prokaryotes. There were these landmark studies on E. coli where researchers extracted mechanosensitive channels and placed them into artificial liposomes. Now, these liposomes are completely devoid of any cytoskeleton or tethering proteins. They're just bare lipid bubbles. And guess what? The channel still opened in response to membrane tension. This unequivocally proved that the lipid bilayer alone is enough. And as we just saw with the Piezo1 nanodomes and similar lipid cork mechanisms in mammalian potassium channels, this brilliant piece of biological engineering has been perfectly conserved across evolution, all the way to human skeletal muscle.
Let's move to Section 3, how Piezo1 maintains muscle health. We're going to focus on Piezo1, which is undoubtedly the star receptor of this explainer. It's essentially a cellular mechanostat.
Let's see how this builds into a step-by-step narrative of muscle preservation. It all starts with physiological loading. Say you're walking or lifting something heavy. That creates membrane tension, which opens Piezo1. This opening allows a transient influx of calcium, immediately converting a physical force into an electrochemical signal. Boom. That calcium spike then triggers the intracellular RhoA/ROCK signaling pathway. Ultimately, this pathway does two huge things. It drives the assembly of the actomyosin network, and it actively suppresses atrophic genes like KLF15 and IL-6. So, a simple calcium influx directly translates to preserving the muscle stem cell pool and actively preventing cellular senescence.
To really anchor this abstract pathway to the bedside, let's think about human biopsy data from patients whose limbs have been casted. When those physical cues from walking or lifting go missing, Piezo1 expression is literally shut off. Without that basal calcium influx, the transcription factor KLF15 just runs unchecked. It ramps up IL-6 and rapidly promotes muscle wasting. It is a perfect molecular-level demonstration of the old saying, use it or lose it.
Which brings us to Section 4, Clinical Therapeutics, Ultrasound Therapy. Because if muscle wasting is caused by a lack of physical activation of these channels, the big question is, how can we as clinicians intervene when a patient is paralyzed, comatose, or otherwise just unable to physically exercise? Well, we looked at clinical therapeutics like ultrasound.
Notice the signal amplification happening here. When we use non-invasive mechanotherapies, the initial stimulus might be very brief, but it gets converted into a massive biological signal through channel crosstalk. A quick pulse of energy hits Piezo1, but it absolutely doesn't end there. That initial little spark cascades into a roaring fire of calcium signaling.
Here is exactly how low-intensity pulsed ultrasound, or LIPUS, pulls this off. Acoustic waves from the ultrasound non-invasively deform the muscle cell membrane, prompting Piezo1 to open and let in a quick calcium spike. This activates an enzyme called PLA2, which generates lipid second messengers known as EETs. These messengers then swing open a second channel, TRVP4, allowing a massive, sustained elevation of calcium. This flood of calcium ultimately activates the calcineurin/NFAT pathway, triggering myogenic differentiation and repair. You are essentially tricking the muscle into a post-exercise repair state without the patient moving a single joint. Finally, we reach Section 5, Muscle as an endocrine organ. We need to zoom out a bit. We've been looking very closely at localized cellular repair, but we really need to redefine how we view skeletal muscle. It is not just an organ of locomotion, it is actually a massive, systemic secretory organ.
This whole process is called mechanico-endocrine coupling. Mechanical load opens surface sensors like Piezo1 and voltage-gated calcium channels. And the crucial point here is that calcium acts as the master switch. It flows in, activating pathways that upregulate a master regulator called PGC1 alpha. This drives the transcription of specific proteins, leading to the vesicular exocytosis of mature myokines, like irisin and IL-6, directly into the patient's bloodstream. Think about that. Local physical training is instantly translated into a whole-body endocrine event.
And this is the breathtaking result. Look at the bidirectional communication stemming from those myokines. Mechanically induced irisin travels through the blood directly to the bone, where it powerfully stimulates osteoblasts to build new bone density. At the exact same time, these signals drive the polarization of macrophages in the immune system from a pro-inflammatory M1 state to a tissue-repairing M2 state. By simply contracting a muscle, you are simultaneously fortifying the skeleton and reprogramming the immune system.
Understanding this systemic crosstalk is exactly why mechanobiology is the future of medicine. The balance of this mechano-mitochondrial axis isn't just about athletic performance, right? It represents a completely novel clinical paradigm. Because if we understand how physical force triggers systemic metabolic and immunological healing, we open the door to targeted pharmacological interventions for complex systemic conditions like type 2 diabetes and osteosarcopenia.
So I want to leave you with this provocative thought. We know the channels, we know the lipid mechanics, we know the exact calcium cascades. If we can perfectly decode the mechanosensitive pathways of Piezo1 and TRVB4, is it possible that in your careers you will actually prescribe an exercise mimetic in a pill? A drug that flips the mechanostat and delivers the full metabolic, skeletal, and immune benefits of a heavy workout to a patient who is completely paralyzed or bedridden. The groundwork is already laid. It's really up to your generation of physicians to bring it to life. Keep questioning, keep exploring, and thank you for joining this explainer.